I borrowed a car. I didn't have a car at that time. Borrowed a car from one of my classmates, drove down to San Jose to one of the big slaughterhouses. I needed eyeballs, and I had a big ice bucket with me.
We'll stop here a moment because there's a lot to unpack. The year is 1982, and yes, Jeremy Nathans is about to negotiate the price of eyeballs with one very confused beef facility manager. But Nathans has a good reason for being at the table. In fact, he's a highly compassionate young vision scientist hoping to use something the industry would typically throw away to lay a foundation that could help vision science realize its ultimate dream.
The goal is to, as best we can, eliminate blindness and visual impairment.
Driving back to Stanford University, Nathans was on his way to an incredible discovery: the very genes that allow us to sense light and color. One of life's defining moments came early for Nathans when his Baltimore high school class visited MIT. Wandering the university's hallways, Nathans was drawn in by a gallery of famous photographs on a wall.
I'm looking at these beautiful pictures. An old man comes out from one of the doors and he says, "Would you like to look at the lab?"
The man turned out to be Franklin Institute medalist and high-speed photography pioneer Howard Edgerton. Nathan's tour of his photo-optics lab sparked a scientific interest that would blossom into a deep appreciation for one of biology's most advanced light-capturing machines, the human eye.
The human eye has special cells called rods and cones. To put it simply, rods have receptors that sense overall light levels. Cones sense the light's color. A person with typical vision has 3 types of cones: one for red light, one for green, and one for blue. These combine to reproduce the millions of shades and colors we're able to perceive.
Because scientists long suspected genetics to be the underlying cause of many vision disorders, Nathans resolved to uncover the genes responsible for the eye's ability to sense light and color in the first place. But in the 1980s, gene identification was incredibly difficult science.
Of course, now we have the complete sequence of the genome. But there was a discovery phase, and this was it, a discovery phase for genetics.
Our DNA is made up of about 3 billion bases. Today, scientists can map the whole sequence in an hour. But decades ago, it took a full day to do just a few hundred. The technology of the time meant Nathans had to do the work by hand.
Locating the genes responsible for light sensing took years, but incredibly, Nathans got the match they were looking for.
We knew we had something. That was a very good moment.
With the genetic sequence in hand for normal sensing, Nathans began researching the genes behind vision disorders. Within just a few more years, his lab announced another groundbreaking discovery. Nathans had located the genetic sequences that make roughly 8% of men and 1 in 200 women unable to distinguish the differences between certain colors. For people who have trouble distinguishing the numbers and the patterns flashing across the screen now, their genes may have one of the sequences discovered in Nathans' lab that causes a variation in the colors their cone cells can sense.
A lot of disorders that have been with us for thousands of years are now accessible in a way that they haven't been ever before, and accessible for new kinds of treatments.
Nathans' career has revolutionized vision science. His work at Johns Hopkins University is helping researchers develop gene therapies and other treatments for visual disorders, including retina implants that may help some regain vision lost to retinal degeneration.
I think there's a beauty to both math and science, an aesthetic appeal. Once you're captivated by it, it doesn't let you go. And I've remained captivated by that beauty.